Hello. This is, I'm tired, Honors Bio Review, and these are my wonderful honors biologists. Say hi. Hi. And a bunch of them did their English project, and they're glad. Yay. Yay. Some have to do it tomorrow, and they're still sad. Yeah. But they're going to get an A, right? Yay. Stranger danger, stranger danger. Yay. Oh, okay. So, first chapter was on enzymes. How did you do with your sub yesterday in the review? Did you work hard?
Oh, yeah.
I only had to yell at like one person on my, what are you doing? Okay, chapter 6, metabolism, energy and enzymes, laws of thermodynamics. Do you know those? What's number 1? Energy cannot be created or destroyed. It can just transfer. And then number 2 says every time it transfers, you lose some as heat. And everything goes to greater and greater disorder. What is that called? Entropy. So if on a scale of 1 to 10, if 10 is the highest entropy possible, what could you say? If I said my bedroom closet right now is a 10, you would say, Miss Litton, that is very disorganized. Good, okay? Now, things will go from organized to disorganized, right? And where do they have more energy? When they have high entropy or low entropy? Where do they have more potential energy?
Low entropy.
Low entropy, good. So things like glucose has low entropy, right? Because it's super, super organized and it has a lot of potential energy in it. Then as it gets transformed through cellular respiration into carbon dioxide and water, we release some of the energy out of the bonds and we use it to make some ATP. A lot of it, a lot of it, a lot of it, or a lot of it, a lot of it. Curious, curious. Why don't we go a lot of it, a lot of it, a lot of it, or a lot of it, a lot of it?
It's like grandma and
ant checks, yes, and biologically speaking, a grandma check is reduced NAD, and an ant check is reduced FAD. And why do we classify them as grandma and ant? The reason is because one gets to go in at the top of the electron transport chain, right? And take advantage of reductase, cytochrome oxidase, which means hydrogen out, hydrogen out, hydrogen out, which means it can come in, a lot of it, a lot of it, a lot of it. But if you're lower energy, okay, just think about like a roller coaster. It can't get all the way to the top. It can only go on the lower roller coaster. And as a result of it, it just gets reductase and cytochrome oxidase. So only 2 hydrogens out means only 2 in, so less ATP. Bueno? Okay. So what are the laws and how do they relate to entropy and energy? We talked about that. What is ATP and its role? ATP is the energy currency of our cells. So reactions that are endergonic will require more ATP. Yeah, they require ATP because you have to put energy into them. Okay, reactions that are exergonic are said to be spontaneous reactions. But they don't just spontaneously break down. We're not falling out of our seats right now and just going into greater and greater disorganization, because even our exergonic reactions are reactions that release energy. There's a little hurdle that keeps everything in check. What is the name of that hurdle?
Energy of activation.
Energy of activation. Who can lower a hurdle for me?
Enzymes.
Enzymes. They are organic catalysts. You could have inorganic catalysts like metals or something that could also do that. Fire, heat, those kind of things do it, but they are not specific. Anything that heat touches, it will lower, right? Okay, but enzymes are specific organic catalysts. Okay, types of reactions, exer, ender, cata, ana. Did I hit all that? Okay. How do enzymes work and how are they regulated? Okay, so we talked about how they work. They have to bind to the substrate. What's the part on the enzyme that binds to the substrate? Okay. So if I were an enzyme and I was going around in my enzymatic reaction and I was joining your hands together, okay, and I'm running around joining your hands together, the active site would be my hands. So if instead of me being able to, all of a sudden these cell phones were in my hands, so now I can't join you together, what kind of inhibitor would you classify this as?
Competitive.
Competitive inhibitor. Perfect. Okay, but if somebody came in and stabbed me in the back, and I'm like, oh, I've got to get this knife out of my back, that would not be a competitive inhibitor. Josh, did you stab me in the back? That would not be a competitive inhibitor. That would be a non-competitive. But it's affecting still my active site, right? Okay. What's something you could give me in the morning that might make me work a little faster? Coffee, exactly. Okay. So what did you say?
Sunlight.
Oh yeah, I'm up before the sun's up, so no. But yeah, the sun, yeah. Okay. Anyway, so there are things that can make an enzyme, like a coenzyme, maybe a vitamin, will make that enzyme work faster, right? To make it do its job. We talked about what's an allosteric inhibitor? I use that word. There's an allosteric site. That's where the knife went in my back. That's the allosteric site that JoJo put there. I want to talk about feedback inhibition. If I'm trying to make a bunch of a product, and I make it, and then when I'm done making, A goes to B, B goes to C, C goes to D, D goes to da. So my product level builds up, and if it comes back and inhibits enzyme 1, okay, then that would be feedback inhibition. Because I don't need to go down this metabolic pathway anymore, because what my end product is, I have enough of it, and it's giving me feedback. Oh, you have enough. Just like feedback, if I turn on my oven to 375, then that's enough heat. It can stop heating up, right? Okay. All right, we did that. 4, what is the difference between being oxidized and reduced, and how does that happen? Okay, which one is about losing energy? Oxidized. Which one's about gaining energy? Reduced. So how can I gain energy? Who will give me energy? Hydrogen. And hydrogen gives me energy by giving me its electrons. So electrons are energy. Hooking up with hydrogen is energy because it always gives you its electrons. So one way to get energy is to hook up with hydrogen, or I could break up with who? Oxygen. Because when I'm with oxygen sucks the life out of me, right? So that's how I can get reduced. I can break up with oxygen, hook up with hydrogen, or gain electrons, gain energy. Those are all reduced states. Now, why did I teach you that before chapter 7 and 8? Because NAD, it's like a switch. It can toggle reduce, toggle oxidize, reduce, it's in one state or the other. If you give NAD or NADP any electrons or hydrogens, you've what it? Reduced it, okay? So in cellular respiration, we take away electrons, hydrogens from glucose, and we give them to NAD. So the electrons that were in glucose are now in NAD. So it's a temporary energy storage. That's why we called it writing a check. We can't spend it, but it is money eventually, right? And then we took that money to the top of the
ETC.
and it dropped off the electrons. Now it lost its value. It's like erasing your check, right? And now it dropped off those electrons. Those electrons are going to go na-na-na-na-na-na. Now those electrons are moving through an electron transport chain. And the beauty of an electron transport chain is that along the way, reductase, cytochrome, oxidase are so excited to have those electrons that it will then take hydrogen ions and take them from the matrix and put them in the intermembrane space for cellular respiration. Okay? And the more hydrogens I have in the intermembrane space, the more they want to come back into the matrix. And when they come back in, they're going to go through what kind of enzymatic? ATP synthase complex. They come back in, push, turn the little turnstile, snap a phosphate onto ADP, and it turns into ATP, higher energy level. Okay? So oxidized is the opposite of that. When NAD dropped off its electrons, it got oxidized. Is it ruined? No, but now it's available to get reduced again. It's the mule. It takes hydrogens from the glucose and drops them off at the electron transport chain. NADP was different, right? In the light reaction, in the second part, we kept the electrons hot. What's that song? Like it's hot. I feel like it's hot. Yeah, something. And so we kept the electrons hot and gave them to NADP, and we reduced it. So we took electrons that got excited by the sun. Oh, where do those electrons come from? Not glucose. Where did these electrons come from? Water. What did we do to the water? Break it in half. So we took away his electrons. We got him charged up, gave him to NADP, and NADP is like, I got the power. Okay? Then it walks from the light reaction into the dark reaction, or the
light independent reaction.
And it brings those electrons, and it's going to give them to carbon dioxide in order to give the energy to carbon dioxide to reduce it into glucose, which has more potential energy, right? Now you put it together?
Yeah.
Oh, I got it now. Okay, and in order to do all those reactions, I need some energy, and I'll use some ATP, which, by the way, we made in the light reaction as well, right? Because we created a hydrogen ion gradient in the photosystem too. PQ makes some ATP, right? Okay, so that's how it happens. That is chapter 6. Do you have any clarifying questions on chapter 6? Yes, sir.
Would you just go over what you said about the light reaction again? Like,
Yes, I will go. I'll do it short now, but I'll hit it again. The light reaction has two parts to it when we do non-cyclic photophosphorylation.
Okay.
Non-cyclic means electrons in and electrons go out. Okay. If it was cyclic, they just spin around and around and around. You wouldn't reduce any NADP. Okay. So in non-cyclic photophosphorylation, we break water in half. When you break water in half, you get two things, oxygens and hydrogens. The oxygens we release and breathe, yay! And the plant can use it for cellular respiration too. The hydrogens donate their electrons to the electron transport chain, and then they're hydrogen ions, because they gave up their electrons, right? So now they're ions, they lost an electron. We can, remember, put them inside the green hollow pancakes, right? Right then. The electrons get excited by the sun, they na-na-na-na. PQ is of electrons that brings hydrogen, even more hydrogen ions in. And then we can use that hydrogen ion gradients when the hydrogens go out of the green pancakes. Then they go through enzymatic, what do they go through? ATP synthase complex, yay, synthesize ATP. So we get ATP from the light reaction, same electrons get excited again, but this time we drop it like it's hot and give them to NADP, who's reduced. And now we've got some check, some money to go build sugar, and we got cash money and we got check money, and we'll go build some sugars.
Okay.
Yes.
Respiration.
Okay.
Come on.
So cute. He raised his hand like I care. All right. So cute. Oh, Dan. All right. Any other questions on chapter 6? Love the Dan. Anything? Anything? Yes.
Reduced has more potential energy.
Yes. Reduced has more potential energy. Anything else, chapter 6? Chapter 6, okay. So, 7 and 8, lay them straight. You need to know the equations for cellular respiration and photosynthesis. You need to identify what gets reduced and what gets oxidized. Do you have that in your notes? Yes, you do. Okay, so cellular respiration, let's do that formula in our head right now. Cellular respiration is C6H12O6. That would be glucose. It is going to get burned, so it's going to get oxidized, right? We're going to release the energy out of it. Glucose gets oxidized. Okay? And then what's another component of cellular respiration? C6H12O6 plus O2. And O2 is who's catching the electrons at the bottom of the electron transport chain. It gets forming water. It gets reduced, right? So oxygen gets reduced, and glucose gets oxidized. Okay? And then for photosynthesis, what do we start out with? CO2, it's going to get reduced in the dark reaction, Calvin cycle light independent reaction, right? CO2 will get reduced into glucose. And then we use water, break it in half, it's going to get what? Oxidized, because we're ripping the hydrogens away from it, right? To create our hydrogen ion gradient and our electron transport chain. Nothing to do. Okay, so make sure you know, and name each major step and where it occurs in its purpose. So let's go through cellular respiration. Why don't we sing it once and then we'll review it, okay? What's the whole thing called? Cellular respiration. What's the first step? Glycolysis. Who does it? All cells. Where do they do it? In the cytoplasm. What do you start out with? Glucose. How many carbons? 6. What are you going to do?
Break it
in
in
half. How many steps?
10.
Enzymes.
10.
What are these called?
2
pyruvic acids. And you reduce
some NAD
and make some ATP. 2. Oh, we have plenty of oxygen. What should we do? Aerobic respiration. What's the next step called? Transition prep. Here we go. And you reduce some NAD and 2 acetyl-CoA, put 1 away. What's the next step called?
Krebs Cycle.
What's another name for it?
Citric acid.
How many times are we going to do it? Twice. Why?
2 acetyl-CoA.
And when we do the bit, we are reducing an NAD and removing a carbon. Here we go. 2 plus 4 is 6. Do the bit, get 5. Do the bit, get 4. Make an ATP, reduce an FAD, reduce an NAD, and 2 plus 4 is 6. Do the bit, get 5. Do the bit, get 4. Make an ATP, reduce an FAD, reduce an NAD, and stop. Gather up all your reduced NADs and FADs and take them to the top of the ETC. Reductase, reductase, cytochrome oxidase, and you make some ATP. A lot of it, a lot of it, a lot of it, or a lot of it, a lot of it. Catch electrons at the bottom. Oxygen. Forming water. Water, 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 water. What are we going to do? Break it in half. What do we get? Oxygen and electrons. Sweep, sweep. Here comes the sun, traveling in waves. Particles of light are called photons, and it gets the electrons excited. Little bunny frou-frou. PQ, and you make some ATP. Here comes the sun, traveling in waves. Particles of light are called photons, and it gets the electrons excited. Who catches it? NADP, forming a reduced NADP. Then you take some ATP and some reduced NADP to the dark reaction, or the Calvin cycle, or the light-dependent reaction. You ready? 3C5. Okay, pause. Who we affectionately know. Right? Okay. Okay. Here we go. Sorry. Stranger danger is like, what is wrong with this class? 3C5 plus 3C1 makes 3C6. Break down to 6C3. Use some ATP. Use some reduced NADP on your 6C3. Take one away, PGAL.
Jazz hands.
And you're left with 5C3. Use some ATP to build 3C5 plus 3C1 makes 3C6. Break down to 6C3. Use some ATP. Use some reduced NADP on your 6C3. Take one away, glucose. And you're left with 5C3. Use some ATP to build 3C5. Stop. Pick up your glucose. How many carbons? 6. What are we going to do?
Break it in half.
Steps?
10.
Enzymes?
10.
What are these called?
2 pyruvic acids.
And you
Reduce some NAD and make some ATP. 2.
Oh no, we don't have any oxygen. What are we going to do?
Fermentation.
How many choices do we have?
2.
You and I make
Lactic acid makes our muscles sore.
Plants and yeast make
Alcohol.
Either way, you oxidize your NADs. Yay! Okay, now, so if we were going to name the major steps, I would talk about glycolysis, right? And that would be the first step of cellular respiration, and I'd say all cells did it. And I would say ultimately it starts with glucose, it ends with pyruvic acid, 10 steps, 10 enzymes. What do I walk away with? 2 reduced NAD and 2 ATP. Yes? The next step I'm going to go on depends if I'm going the aerobic pathway or the anaerobic pathway. If I'm going the aerobic pathway, my next step is transition prep. I start with pyruvic acid and I end with what? 2 acetyl-CoAs, and I also get 2 reduced NADs. Do I make any ATP in that step?
No.
I do not. The next step I'm going to do is the Krebs cycle, and I'm going to put each of the acetyl-CoAs through the Krebs cycle. And I'm going to start with 2 acetyl-CoAs. That's 1, 2, 3, 4 carbons, right? They're going to come off every time I do the bit. And I'm going to walk away with 1, 2, 3 reduced NADs, right? 2 times around means 6. Okay, and 1 FAD a turn, so that means 2, and 1 ATP a turn, which means 2. When I am done with the Krebs citric acid cycle, there are no more carbons left for me to take any energy out of. Okay, now all I have is my checks to cash. Okay, I either put them at the top or partway through my electron transport chain. They drop off their electrons. It empowers receptors to go, wee, reductase, cytochrome oxidase. I create a hydrogen ion gradient. I use that hydrogen ion gradient to synthesize ATP. The bulk of the ATP synthesis takes place in the ETC. The bulk of the formation of NADs happens during the Krebs cycle. Now, if I have no oxygen and I have to go down the anaerobic pathway, I still do glycolysis, everything that I said, reduce some NAD and make some ATP too. The only reason you do fermentation is you have to take the NADs you reduced and you have to oxidize them again so you can reduce them again. Okay, I have to have a way to oxidize them. We use the electron transport chain to oxidize them, right? Take them to the top, so ETC, I need a okay? And I don't have an electron transport chain to do that. So I will run out, I have a finite amount of NAD, I will run out of NAD to reduce, and if I can't reduce NAD, then I can't make any ATP, okay? That's why you do fermentation, is to oxidize the NAD. And what you change your pyruvic acid into is either going to be lactic acid or alcohol, and it just depends on what kind of metabolic pathway your body has, your living organism has. Okay, good with cellular respiration? Yes?
There's one question on the ETC. So, like reductase and cytochrome are both where hydrogen ions go into the mitochondria to make the gradient, right?
Yeah. They go from the matrix of the mitochondria, the inner chamber of the mitochondria, into, hi sweetheart, the intermembrane space.
Okay, and then what does oxidase do?
Reductase, cytochrome, oxidase. Those are both receptors that go kick and move it.
Did you find car keys?
I did not, sweetheart.
Oh, look at that.
Look in your box of chocolates.
I did.
Oh, okay.
Do you want any more though?
No, you already bought 5. Okay. All right. Oh, glad we got that. Okay. Can I answer your question?
Yeah.
Okay. Any other questions about cellular respiration? All right, so let's talk about photosynthesis. Photosynthesis has 2 major pathways, light reaction and the dark reaction, comes in light interaction, right? The light reaction, its job is to start with water, right? That's its starting point in sunlight, right? And it breaks the water in half. And the sunlight doesn't do that. There's an enzyme that does that. Its name is enzyme C. I just thought I would mention that. Okay? And it breaks the water in half. And when it does that, you have oxygen, super good for us. You don't have, I didn't put on the test, I'm just telling you. I have oxygen and I have hydrogen, because those are 2 things that make up water. H2O, okay? The oxygen gets released. The hydrogens are made out of, all hydrogens are made out of 1 proton and 1 electron. So it gives up its electron to go right? It gives it up for that, and the hydrogen ions themselves get brought into the green hollow pancakes, the thylakoid membranes, right? Remember, we have different pigments and they absorb different wavelengths of light. Not all colors of light are equal to a plant, right? It likes some colors better than others. What would you say is its least favorite color?
Green.
I agree, okay? You put it in green light, you might as well be putting it in the dark, okay? So then the pigments absorb the light, get the electrons excited to a high energy level. They get passed from receptor to receptor. At one of them in particular, it's able to crank some more hydrogen ions inside the green hollow pancakes, and his name is PQ. PQ, right? And then take the same electrons, excite them a second time, and then drop them like it's hot into NADP, reducing the NADP. So now we have high energy, reduced, lots of potential energy and reduced NADP. We have actual ATP that we made from photosystem 2, and we do photosystem 1, okay? And we're going to take those to the dark reaction, Calvin cycle light independent reaction, and that occurs not in the pancakes and not on the pancakes, but outside of the pancakes, right? In the stroma, the syrup, okay? Out in the stroma. And there in the stroma, you're going to do your dark reaction. You have carbon dioxide now, one of your reactants comes into play. And CO2 hooks up with our fave, RUBP. She's ready to party, okay? And RUBP and CO2 hook up, but it's always unstable when you hook up with her, remember? So that RUBP is a C5, CO2 is a C1. When they get together, they are a C6, but they break in half. And we're going to do 3 at a time, you know, so we have a 5 and a 1 makes 6, break it in half, 5 and 1, break it in half, 5 and 1, break, right? So now we have 6 C3 units. We throw some electrons at them using some reduced NADP. We throw some energy at them using ATP. And we make high energy half of a glucose called PJL. And another name for PJL is G3P. Okay? And so then we use that, and we could stop right there and walk away, right, with 3 glucoses since we have 6 halves. But we want to keep the party going. So we take how many away? 1. Leave 5 there. Rearrange them using some ATP to build 3C5 to regenerate our UBP again so she can keep partying with some more CO2. Right? Goes around 3 at a time, right? 3 CO2s at a time. These 3 CO2s go in as individuals, they come out as a half of a glucose. Another 3 go in as individual CO2s, they come out as a half a glucose. Then we put those halves together, we have a whole glucose if we want it. Or we can build fructose, or an amino acid, or a fatty acid, or starch. We can build whatever we want with it, those halves of glucose. Now, what can go bad? Photorespiration. That's a clear amalgamation of 2 different metabolic processes, because it sounds like photosynthesis, but it gets hijacked in the end, respiration. And the reason is RUBP is not faithful, and she will hook up with who? Oxygen instead of carbon dioxide. And when she does that, it screws the whole cycle up. It's not photosynthesis, it's not cellular respiration, it's not helpful at all. So to take care of her unfaithfulness, 2 different strategies. C4 plants hide her away, surround her with mesophyll cells, and a compound called PEP, who is always faithful. PEP hooks up with carbon dioxide only and escorts the carbon dioxide into RUBP so she's never tempted by the fruit of another. That's one way to do it. Okay, that's a partition in space, right? It's like, here, you just do your dark reaction here. And those are C4 plants. C4 plants. Most plants in the world are C3 plants. C4 plants are those. Yes?
So they're like the protective parents?
Yes, they're like the protective parents.
Like hide their together?
Yes.
That's all I need.
That would be a way to remember it. Okay, and then there's another one. It's called CAM plants, C-A-M, all caps. CAM plants are like desert plants, and at night, you know, is the only time when they have their stomata open because it's too hot. Damn, those CAMs, it's just so hot, okay? And so they will fix CO2 at night with their PEP, and then when their stomata are closed during the day, they will release it slowly out to RUBP and dish it out so that she's never tempted by the fruit of another. Yes? And then the question is, do you know your additional objectives?
Pretty well.
Okay? Because will they be on the test? Yes, they will. Yes, they will. And so when we look at and compare and contrast them, okay, because we would know that's a chloroplast and that's mitochondria, right? You can compare and contrast those 2. What's something they have the same?
So they all
What?
produce ATP.
They all make ATP. Good. What else?
They have the same molecules in both.
Yeah, what do they have?
H2O and
Okay, but their reactants and products are different, so we better be careful on that. What do they both have? They both concentrate hydrogen ions. They concentrate them. Where does cellular respiration, when you do cellular respiration, where does he concentrate them? Reductase side of he in the intermembrane space, right? Where do they? Inside the thylakoid, so the interior. Okay, what else do they do? They both have cycles, right? What's the name of the cycle in Krebs? Oh. Okay, and in the Krebs cycle, you're kicking off CO2, right? But in photosynthesis, what's the name of the cycle? Calvin, or a bunch of other names. And in this one, you are doing what? Introducing CO2, right? Okay, they both have their own DNA, right? Own DNA. What do we know about that? Own DNA, own ribosomes, they're double membranes. Yeah. What kind of energy do we primarily produce, energy-wise? Yes, I think I would put here, energy. Okay, so on this one, energy, we're making a bunch of it, and it's called what? Yeah, ATP. ATP. We make it. I don't like the way I did that. We make it. Okay, and the energy over here, we have what? What is it? What do we use? The sun, and we have to use it, right? We have to use the sun in order to ultimately make our sugar. All of a sudden, that got traction. Okay? Yes.
Do some class lab questions or
What?
do some class lab questions.
You know what? I don't have any put in here right here. I'm sorry. But do the where you know what would be really good questions for you to do? The ones at the end of the chapter. And where are your answers to that?
Back in the book.
Yes, because those are the ones I tend to use. Ones very much like that. Okay. Any other questions or concerns? Yes. Wait, I can't hear. Shh.
CAM plants also have PB and
PEP.
PEP.
Yes. PEP for them works at night, storing the CO2 as a solid, whereas C4 plants, they isolate the dark reaction, the calcicyclide independent reaction. You're welcome, sweetheart. And then PEP escorts it in. Yeah. Yes.
The transition also produces 2 carbon dioxide. Is that one?
Yes. Yes. Transition also produces 2 carbon dioxide. Exactly. Okay, you good? You good? You good? Okay. Get rest. You got this. I'm already proud of you. Have a piece of toast if it's late. Don't stay up too late. Know that I love you. Make good choices.